Others will be on YouTube only.
Yes, sir.
तो वो लोग यहाँ पर दिखाई देंगे या नहीं देंगे?
यस सर, देंगे सर। नो सर, वो लोग यहाँ पे नहीं, वो सर यूट्यूब पे बस। हेलो मैम।
हाँ
ज्वाइन कर ली।
हाँ बोलो।
मैम स्मृति मैम को कॉल था।
ओ ठीक है। हम्म।
सर मैं अपना वीडियो ऑफ कर दे रहा हूँ। गुड मॉर्निंग मैम।
गुड मॉर्निंग।
Good morning, sir.
Good morning, sir.
Sir, you are in a new look, sir, after a long days.
Yes, sir. With beards.
Sir. Participants are in YouTube, sir.
Huh?
Participants are on YouTube.
Okay.
Good morning, sir.
Good morning, ma'am. How are you?
I'm fine, sir.
राजकुमारी मैम नहीं होने से सर कॉन्फ्रेंस आधा हो जाए, एकदम आधा.
That is true, sir. She brings lively to the any of the event which has been organized.
Yes, sir. वो मतलब 50% तो
Thank
पहले
you,
से हो
sir.
जाता है.
It's an honor to me.
No, ma'am. So, sir, how was that effect of the cyclone? South, sir? Sir, how was the effect of the cyclone over there in
No,
Odisha?
sir, not at all. Only electrification and internet is affected. Otherwise,
Okay. Otherwise, it is okay. I think this time the Bengal has the maximum effect.
Bengal had a horrible condition.
ज्यादा सर इन्फ्लुएंस है बालासोर के आगे साइक्लोन का.
Cyclone. Balasore से आगे.
और यहाँ पर जितना भी है आप लोगों को लगेगा, लेकिन हम लोगों को नहीं लगेगा. Because every year
You
we
are
face that.
habitual of that. Mostly cyclones are faced at the Odisha border only.
Every year Odisha is affected.
Has one or the other.
And this year probably the cyclone has given us a warning. तो इस बार छोड़ के जा रहा हूँ, थोड़ा अच्छे से चलो वरना देख लूंगा. अभी भी सर लाइन नहीं है, वो इन्वर्टर से करना है. हाँ सर, अभी भी लाइन नहीं है. इलेक्ट्रिफिकेशन इज नॉट देयर.
अच्छा, इलेक्ट्रिसिटी नहीं है. तो आप कैसे आ
गुड
रहे हैं?
मॉर्निंग सर. हाँ सर, गुड मॉर्निंग सर. अंगद सर आ गए.
गुड मॉर्निंग. हाँ, गुड मॉर्निंग सर.
Good morning, Angad sir.
गुड मॉर्निंग मैडम. गुड मॉर्निंग.
गुड मॉर्निंग सर.
हाँ, गुड मॉर्निंग सर. गुड मॉर्निंग सर.
अंगद सर तो और यंग हो रहे हैं।
हाँ। अरे सर, यही तो, जब 2 महीने बाद हम लोग मिलेंगे तो कौन क्या हो रहा है पता चलेगा।
हाँ।
Sorry is always smart.
अमित जी ज्वाइन कर लिए?
नहीं आए अभी तक।
जी सर, अभी
नहीं
नहीं
आए
आए
अभी
नहीं,
तक।
आए नहीं।
उनका तो पर्व भी आ जाए थोड़ा।
Sir, all credit goes to Prashant
Yes sir.
for this e-FDP.
प्रशांत जी,
Prashant is doing all the smart works in this e-FDP. Sir.
I was just about to say, very well done. I was just looking at it. So nicely it is done.
तो आप जहाँ पर हो तो सब अपने आप हो जाएगा मैम।
नहीं सर, ये आप लोगों का एफर्ट, पूरा एफर्ट है
सर विदाउट
डॉक्टर का।
इंस्पिरेशन, आप लोगों के बिना इंस्पिरेशन से एक कदम भी आगे बढ़ना मुश्किल होता है।
अरे नहीं सर, आप लोग जो अच्छा कर रहे हैं उसमें सब हम लोग साथ रहते हैं हमेशा।
दिस इज वन ऑफ
बट
द,
जो भी भूल चूक होगा सर,
अरे
पहले से माफी मांग लेता हूँ।
कोई भूल चूक हुआ ही नहीं है, ना होगा। अच्छा काम में कहीं भूल चूक नहीं होता है सर। Everything is okay। सब ठीक है, आप अच्छा कर रहे हैं। इस बीच हमारे टीचर बहुत किए हैं सब।
थैंक यू सर। जेएमसी का तो आज से फिर सीरीज ऑफ वेबिनार्स चल रहा है सर।
इसका
जेएमसी का राहुल सर,
राहुल सर।
हाँ हाँ हाँ।
पूरा वीक भर दिए हैं सर। गुड मॉर्निंग अखिलेश सर। प्लीज अनम्यूट योर ऑडियो सर। सर अखिलेश प्रताप सिंह सर जी आए हैं अभी, जो हमारे ऑल इंडिया फार्मा एजुकेशन सोसाइटी के प्रेसिडेंट हैं।
गुड मॉर्निंग
वेलकम
सर।
अखिलेश जी, गुड मॉर्निंग एंड वेलकम सर।
गुड मॉर्निंग।
बिल्कुल हो रहा है अभी क्लियर। I'm properly connected or not, sir? Your voice is clear here.
सर प्रशांत सर वीसी सर इन किए हैं क्या?
He has not yet entered, sir. No.
मैं तो 2 बार कॉल कर चुका हूँ सर की पिकअप नहीं कर रहे हैं। एक बार और कॉल करें।
जी जी, एक्चुअली ये बोल रहे हैं कि हमारी क्लियर हो रहा है, फेस वगैरह विजिबल हो रहा है, क्लियरिटी? हाँ, विजिबिलिटी क्लियर है। जी जी, फेस वगैरह क्लियर विजिबल हो रहा है अभी? एक्चुअली इधर शो नहीं कर रहा है ना कि मेरा विजिबल हो रहा है कि नहीं हो रहा है। अच्छा आप देख, अच्छा आप ही देख पा रहे हैं?
हाँ, हेलो। रजिस्टर सर, VC सर इज नॉट लिफ्टिंग फोन सर।
ओके,
उनका फोन बज रहा है, 3-4 बार बज रहा है।
वो भी आज पर्व है सर, हो सकता है कहीं नमाज में गए हो।
हाँ,
कैन आई टेक योर हेल्प?
बट अभी मॉर्निंग के टाइम में तो हो आए होंगे।
मे बी नमाज टाइमिंग अप्रोक्सीमेट बात 12 PM अप्रोक्सीमेट 12 PM।
मैं एक बार ट्राई करता हूँ।
तो सर काइंडली करें BC सर also।
प्रशांत सर, डायरेक्ट सर इन किए क्या?
Let me check it. Oh, no sir.
सर लेट हो रहा है, क्या करेंगे?
स्टार्ट सर।
और 2 मिनट इंतजार करेंगे। सर क्या करेंगे? संगत सर,
इसी सर में लेट होगा ना।
हाँ सर, सबके भी
स्टार्ट कर दीजिए ना सर, स्टार्ट कर दीजिए, उसके बाद आएंगे तो सर का बाद में हो जाएगा।
गुड
सभी
मॉर्निंग
लोग
सर।
इन हो गए?
हाँ, बाकी सब यूट्यूब में है सर। पार्टिसिपेंट सर ऑन यूट्यूब।
यूट्यूब ना।
और गेस्ट रेडी है सर ना?
हाँ
अपने स्पीकर रेडी है ना?
हाँ सर।
यस यस।
तो आप स्टार्ट कर दीजिए, VC सर आएंगे तो उनका फिर बाद में ही कर दीजिए, लेट हो रहा है। स्टार्ट।
स्टार्ट करेंगे ना? प्रशांत
सर
स्टार्ट.
ओके सर.
Yes, ma'am.
Start, sir. Hello.
Yes, we can.
Okay, I'll begin then. A very good morning to one and all. Today it gives me great pleasure on this virtual platform on behalf of the School of Pharmacy to extend a very warm welcome to the dignified guest, Mr. Akhilesh Pratap Mishra, the President of IPES, that is Indian Pharma Educational Society. I also take the privilege to extend a very warm welcome to our Vice Chancellor, Sir of Arka Jain University, Dr. S.S. Razi. On this platform, I also take the opportunity to wish our VC Sir Eid Mubarak. May the dark clouds of today pass away with the wind of time and bring a bright, sparkling Eid in 2021. I also welcome our Director, Sir, Mr. Amit Srivastava, our CFO, Mrs. Richa Srivastava, our Director Campus, Dr. Prashant Tiwari, Registrar, Sir, Mr. Jasbir Dhanjal. In fact, I welcome the entire body of management of Arka Jain University. I also welcome all the deans, program coordinators, and the entire faculty fraternity to this e-faculty development program. Future challenges, possibilities, and emerging trends in vaccine and drug development for viral diseases. This program is actually jointly organized by School of Pharmacy, Arka Jain University, and Indian Pharma Educational Society, IPES. We all know that light and brightness has always been equated with motivation, harbinger of good, and associated with spiritual being. It is a tradition to light a lamp first before starting any auspicious events or rituals. A lamp teaches to be upright, rise upwards, and dispel darkness. Knowledge removes ignorance, just as light removes darkness. Hence, we light the lamp to bow down to knowledge as the greatest of all forms of wealth. Similarly, we should acquire such knowledge as to take us towards higher ideals. A single lamp can light hundreds more, just as a man of knowledge can give it to many more. The brilliance of the light does not diminish despite its repeated use to light many more lamps. So true. Knowledge does not lessen when shared with or imparted to others. To begin with the program, I request Dr. Prashant to get ready with the lamp lighting ceremony. Dr. Prashant, are we ready?
Yeah. Please proceed, ma'am.
So, I request all the dignitaries to come up for lighting of the lamp. Of course, once you are the company dean of School of Health and Life Sciences. Yes. Prashant, please continue.
Yes. Thank you.
श्वेत कमल तेरा आसन है, श्वेत हंस तेरा वाहन है, हम पर स्निग्ध दृष्टि कर जननी बोध क्षीर की धार दे। तेरा वाणी तार दे, तार दे, तार दे, माँ सरस्वती शारदे, माँ सरस्वती शारदे। इस अज्ञान उदय से, इस अज्ञान उदय से, वीणा वाणी तार दे, तार दे, तार दे, माँ सरस्वती शारदे, माँ सरस्वती शारदे।
Thank you so much, Prashant.
Thank you, ma'am.
Okay, so shall I continue?
Yeah.
Okay. I just want to have a talk with our guests. Sir, have you ever come to Jamshedpur? Akhilesh sir,
Yes, ma'am.
you come to Jamshedpur?
Yeah, what actually your voice is not clear here, so can you repeat?
My voice is, now is it clear?
It's clear now.
Am I audible?
Yes, audible, ma'am.
Okay,
Okay.
sir, I was just asking, have you ever come to Jamshedpur?
Yes, welcome.
No.
What you want to say?
He's asking,
Okay.
आपने इसके पहले बिलासपुर जमशेदपुर आए हैं क्या कभी इसके पहले?
No, actually, अभी अभी तो विजिट लेंगे, नेक्स्ट टाइम विजिट करते हैं.
Okay.
What, even I couldn't hear what sir said. Did he come to Jamshedpur?
No, he didn't came.
Oh, he did not come. Okay, so sir, right now what we have, it is exclusively for you. We will take you to a trip to Arka Jain University. And also, like, I have a friend COVID. So I thank my friend to make us so techno savvy. So virtually, I'll be taking you to a virtual, to visualize our Arka Jain University. So Prashant, can we have the trip to AGU?
Yes, please.
And this is exclusively for our guests. We will all enjoy, but this is exclusively for our guests. So Prashant, let us have that.
Yes.
Whether it is school, higher education, or postgraduate or entrepreneurship, the discovery of mind is very important. Learning should be fun, learning should be creative, and we should try to assimilate thoughts in such a way, and this is where the youth is empowered here to see that he looks forward and tries to contribute not only for himself but to the society as a whole.
Arka Jain University, Jamshedpur, came into existence about a year back, that is 2017. I hope and pray that this Arka Jain University, which came into existence about a year back, will be a center for excellence in the days and years to come, not only in India, but in international level.
Hello everyone. So Jain College in Arka Jain University is a fascinating story. It's a story of basically ordinary people doing very extraordinary things. In a very small classroom, that the journey that we are starting with 34 students will reach here, that we will be conducting our commencement day in the biggest auditorium in the city. And that's how today we are here. So entrepreneurship is the DNA of the Arka Jain University and the JGI group. If you look at the JGI group, we have incubated more than 100 companies, which has a turnover of more than 4,000 crore.
In Arka Jain University, we want to focus on the students and the teachers. What we feel that when students come into the campus, then they should get a unique experience. We have produced many sports personalities.
We have well-equipped library like computer lab, engineering lab, fashion designing lab, mass comm lab, and bioscience lab facilities for the students to enhance their practical knowledge.
Without setting the goal, nothing is possible. Kindly I request you, first of all, set the goal and try to achieve it. Arka Jain University tried to help achievement to your goal and provided highly qualified and experienced faculty.
One of the most important phase of every individual is the college life, where a teenager drifting into the ocean of new beginnings and possibilities.
I'm studying here since 1 year and I had a great experience studying in this university. I'm going to talk about the stereotype, how people think that girls cannot take admission in mechanical or electrical branches because it's totally made for boys. And this university broke this stereotype by giving admissions to all the students as girls or boys. They have equal opportunities for studying in all the branches.
Hello, here Dr. Jyotirmay Sahu, Dean of School of Pharmacy, Arka Jain University. School of Pharmacy is trustworthy. You will definitely like the lovely campus in a pleasant environment with world-class infrastructure, well-equipped laboratories, library with large range of books and journals, highly qualified faculty. You are one of a kind, so are
Student of School of Pharmacy of this university. We are the first batch of B.Pharm of 60 students of this university, but we feel no such pressure of the first batch because of the constant guidance and support of our faculty. And the best part which I feel is that we have a separate library for our department only with the best publications.
About my college, I would just like to say you that it's just not a college, it's something beyond it. Here with good education, you will learn moral and ethical values which will help you to achieve your goals.
Okay, that was excellent. I hope it's over, Prashant.
Yes.
Okay, excellent. That was. I hope our guest has also enjoyed with us.
Yes, ma'am. Because the video session was excellent.
Oh, sir, thank you so much. It's an honor for this type of compliment from you.
Okay, thank you.
Prashant, do we have our VC, sir?
I think yet not, sir, has came.
Okay, sir. Then, Jyoti, sir, shall we continue with the next one with your one overview? Hello, am I audible? Jyoti, sir?
Just I would like to request Angad sir to say few lines.
Okay. So, Angad, sir, may we welcome you to address the gathering for today? Am I audible, Angad, sir?
Yes.
Okay.
हेलो, आवाज आ रही है सर?
Yes, sir. आ जा रहा है.
Yes, sir.
Welcome, Dr. Mishra, and lot of thanks, sir, for sharing the valuable time with the Arka Jain University, and thanks to Dr. Shahu and his faculty member. Hello, आवाज आ रही है सर?
Yes, sir. Please proceed.
आवाज कट रही है. Most welcome to Dr. S.S. Raji, Vice Chancellor, and a special thank to Mr. Amit Kumar Srivastava, Dr. Jashbir Dhanjal, and the Finance Officer, Mrs. Risha Garg. Sir, hello.
Yes, sir.
Yes, sir.
Are you audible, sir?
Yes, sir. You are audible, sir.
Voice is properly clear.
Hello.
Yes, sir. You are audible, sir. We can hear you.
Dr. Mishra, welcome, sir. Lot of thanks for the sharing the valuable time with Arka Jain University, and best of wishes for the faculty development program, sir. Thanks to everyone for the giving the time.
Now, I request our Dean of Allied Science, Health and Allied Science, Dr. Jyotirmoy Shahu of Arka Jain University, present the overview of this e-FDP.
Thank you, ma'am.
Sir, over
Good
to
morning,
you.
one and all. Good morning, one and all.
Good morning, sir.
Good morning.
Good morning, Akhilesh. A warm welcome to all delegates, respected faculty participants from different institutions and dignitaries to the 6 days e-FDP program, Future Challenges, Possibilities and Emerging Trends in Vaccines and Drug Development for Viral Diseases. Jointly organized by All India Sharma Educational Society School of Pharmacy and IQAC of Arka Jain University. I am thankful to the Honorable Chancellor, Mr. of Arka Jain University, Dr. Raichand Chandra sir, President of AIPS, Mr. Akhilesh Pratap Misraji, Vice Chancellor of Arka Jain University, Dr. S.S. Raji sir, respected Director, Mr. Amit Srivastava ji, Registrar, Mr. Jashbir Dhanjal sir, Dean Student of Welfare and Director of Campus, Dr. Angad Tiwari sir, and Chief Financial Officer, respected Richa Madam, for the kind support to conduct this e-FDP. My heartfelt gratitude to Mr. Bhupender Kausik, Product Development Leader, R&D, Martindale, United Kingdom, London, for sharing a session with us today after this inauguration session. I am thankful to other eminent resource persons, Professor Dr. Pratap Kumar Sahu, School of Pharmaceutical Sciences, Shiksha Anusandhan University, Bhubaneswar, Dr. Santosh Sharma, visiting faculty at IIM, IIT, Arka Jain University, Jharkhand, Professor Dr. G.P. Mahanta, Head Department of Pharmacy, Annamalai University, Tamil Nadu, Professor Dr. Erwin Martins Faller, School of Pharmacy, San Pedro College, Philippines, Dr. Nilesh Meher, Naipur, Hyderabad. Dr. Sachin Kumar Singh, Department of Pharmacy, Lovely Professional University. Dr. Pankaj Kumar Singh, Naipur, Hyderabad. Dr. Harish Chaudhary, MBBS, Consultant Critical Care Medicine, Amri Hospital, Bhubaneswar. Dr. Devendra Kumar Devangans, Site Head Formulation Technology Transfer, Cipla Limited. Dr. Dinesh Kumar, Material Science and API Specialist, SK Biotech, Shorts, Dublin, Ireland. Who will share the session in this 6 days EFD? It's really a time for self-analysis that after 8 months of outbreaking of pandemic COVID-19, the world is not able to present an antidote even in 21st century. Yes, it does not mean that we have not done anything against viral infections. Vaccines have transformed the landscape of viral diseases during last century to save the lives of millions of people worldwide. As a result, there is complete elimination of smallpox, poliomyelitis, measles, mumps, rubella, chickenpox, yellow fever, rabies, hepatitis B, and also there is development of vaccines against rotavirus and papilloma viruses. Despite of these remarkable success, we do not have vaccines against HIV, HCV, RSV, and at present, entire world is struggling against COVID-19. The most challenging part of development of vaccines against viruses are based on structural understanding of the interplay of neutralizing antibodies and viruses. Development of antiviral drugs is too challenging. It involving many stages like target identification and screening, lead generation and optimization, preclinical and postclinical investigations. In spite of modern tools and stringent quality control measures, only a few antiviral drugs are getting approved for human use, maybe due to side effects or antiviral drug resistance. Increasing knowledge about viruses, mechanism of their infections, and the rapid evolvement of novel antiviral strategies and techniques will definitely speed up development of antiviral drugs for which today we are waiting. Education needs to build up the nation. Our top priority is sharing of knowledge to ensure the future. This is a wonderful platform where resource persons from different parts of the globe will share their knowledge within this 6 days of e-FDP. Hope you all will and we all will enjoy this. This is because we believe sharing of knowledge will fulfill the need of education system and can ensure our nation as well as future. I wish for the great success of this e-FDP, and I am thankful for your whole cooperation, all the participants here. Thank you. Stay home, stay safe. Thank you, ma'am.
Yes.
Thank you so much, sir. So, moving on to the next. May we request the president of IPES, Mr. Akhilesh Mishra, to address today's virtual gathering. The mic is yours now.
Yeah, thank you very much, ma'am, for giving me this opportunity. So, first of all, very good morning to distinguished guest Dr. S.S. Raji sir, Chancellor, Kajari University, and Dr. Jyotirmaya Sahu, Dean's School of Health and Allied Science and convener of e-FDP, and Dr. Sweta Tripathi, coordinator of e-FDP, and organizing secretary of e-FDP, Dr. Prashant Tiwari, and all respected faculty participants. It is my pleasure to welcome you all on e-faculty development programs on future challenges, possibility, emerging trends, development or viral disease. This e-FDP will help teachers to strengthen themselves in the pharma educations, innovations, awareness, motivations, event culture of the respective college. As it will create awareness about pharma professional development. This will ultimately have a positive impact development of the participant college. So I am briefing about the IPES. So I involved in assessing skill-based quality education, digital learning, correction, and most appropriate practical applicability in the pharmaceutical professions. So IPS committed to fulfill of the need of the student of the professional at single place, like the certifications, appreciation, advancing support, fellowship, awareness, job assistance, training, competitive exam preparations, sponsorship, event and assisting organization for scientific conference, seminars, workshops, and other educational and technical events. So we are indeed grateful gracious presence and session on behalf of Indian Pharma Educational Society. I would like to extend a gratitude and appreciation to Dr. S.S. Raji, Vice Chancellor, AG Jamshedpur, and Dr. Jyotirmaya Sahu, Dean's Faculty of Health and Applied Science and convener of FDP. So I'm very thankful to Dr. S.S. Raji and Dr. Jyotirmaya Sahu to organizing and participating in this college and delivering a wonderful session and express and address all participants and also the other. So I am delivering and the society. So after the session of this sessions, society would like to extend a gratitude and appreciation to all our invited speakers by lifetime membership of IPS and a certificate of appreciation. And I am very thankful to Dr. Prashant Tiwari and all who are directly or indirectly involved to support and help to organizing e-FDP. So hope this collaboration of e-FDP and AJU will organize different pharma test or campus virtually for the development of the pharmacopoeians. So we are very happy and about the speakers and other professionals. Dr. Jyotirmaya Sahu already discussed about the speakers and or resourceful person, and their topics also will be discussed already were discussed by Dr. Jyotirmaya Sahu. So I am very thankful to entire teams of Alkazan University and Dr. Prashant Tiwari, and Marine Secretary, and Dr. Sweta Srivastava, for coordinating this event. So hope all of participants cooperate this successful event of in the pharmaceutical profession. So thank you all the best for the full development of the SKIP of the FDP faculty development program. Thank you very much.
Thank you so much, sir. It was so nice hearing from you. We also love to hear from our registrar, sir. So we just cannot let him go like that. Sir, we would like you to share few words with us on this virtual platform now.
Yeah, warm welcome to each and everyone who has joined this, and especially to Mr. Akhilesh Pratap, who has basically spent us with some of his valuable time so that FDP for the faculty development could be taken place along with the support of Dr. Sahu and Dr. Prashant Tiwari. And I would take this opportunity to even thanks our director campus who is there and available with us from his busy schedule. So once again, a warm welcome to each and every one of you. Thank you, ma'am.
Thank you, sir.
Okay.
Hello.
Yes, sir. Yes, sir. Thank you so much, sir. It was so nice hearing from you. Now we move on to the presentation of lifetime membership to Honorable Vice Chancellor, sir, by the President of IPES. Sir, can we have this now?
I think everybody should have a clap for our Vice Chancellor, sir, on their virtual.
Yes.
Yes.
Wherever they are, please have a clap.
ताली मार बाकी कुछ ये
That's done. Okay. Coming to towards the end of this first session, we request everyone wherever they are to stand up for the national anthem.
Jai Hind.
Dr. Prashant, that's done? Is it done, Dr. Prashant?
Hello,
Your
Dr. Prashant.
mic is on mute.
Yes, it is done.
Okay. Thank you so much. I have still not said. Thank you so much. In fact, thank you all the guests who are present here. We are really very sad that we couldn't offer high tea to you. It's already 11:17. We couldn't offer this, but this is a due from Arka Jain University. We will all meet one day and we will have a fantastic high tea. So now I request, this is the coming towards the end. This was the end of session 1 for day 1. Now I hand over this mic, this imaginary mic to pharmacy department to continue with the program. And that was all on my part that I had to do. Okay, Jyoti sir?
Thank you, madam, for your kind cooperation. And we expect the same in future from you. A intimate cooperation, always be with School of Pharmacy and School of Health and Allied Sciences. Thank you, madam.
Thank you so much, sir. And all the good wishes. Now is it over to Smriti? Smriti is going to conduct?
Yes, ma'am.
Okay, all the best, Smriti. Continue.
Thank you so much, ma'am. Good morning, everyone, to this virtual platform of 6 days e-FDP. I would like to introduce to you all Mr. Bhupendra Kaushik, who is a product development leader, Martindale, London. He is an accomplished pharmaceutical researcher over a decade in global pharmaceutical research organization with particular interest in new drug development process and commercialization. He has handled new drug product development projects for a variety of drug substances from product design stage through to first time clinical supplies and industrialization. Most of his experiences comes from leading scientific frontiers at GSK research and development units based around London. He has been a part of successful product launches and process validation of first commercial product on continuous tableting line. He has also led product asset swap between Novartis and GSK as a part of business divestment. He is a sought-after speaker on many international seminars, conferences, and workshops. Moreover, he is a lifetime member of Royal Society of Chemists and honorary lecturer in several universities. It gives me immense pleasure now to invite Mr. Bhupendra Kaushik sir for his session on drug development for viral disease. Over to you, sir.
Thank you so much. That was really great. Thank you. Let me share my screen and let me know when you see it, please.
Yeah.
Do you see it?
Yeah, sir. Please.
Okay, just a second. And can you hear me clearly?
Yeah, sir.
Perfect. First of all, I would like to thank everybody who has been part of this session today, especially the participants and the organizing committee. I would also like to express my gratitude to my dear junior Prashant, who has been working really hard to make it possible. I'm also very thankful for the organizing committee, the senior leadership, and dear principal that they have offered me this opportunity to share my experience with the rest of the team. I hope everybody who is joining in today will benefit from this session. Can I please request everybody to mute their devices so we can hear this presentation clearly?
Yeah, sir.
Thank you so much. I can still see a few people. Can I please request?
Okay. I'm doing, sir. Please proceed. No worry.
Okay, sure. Thank you. Thank you so much, Prashant. Let's start this journey, okay? And I use the term journey because this is a journey of a molecule which gets discovered sometime accidentally, sometime intentionally, sometime using a structured approach. And this journey starts from smaller studies in animal and progresses to human. And if is identified as one of those molecules that could be a bigger commercial success, then one day it becomes a blockbuster. And when I look at my sort of journey, personal journey, let's start with my educational qualification. I hold a bachelor degree in pharmacy, and I hold two different master degrees, one from India and one from London, actually. My actual working experience started back in India at MCure Pharmaceutical, working on oncologic injectable products. And my first work experience in London started with a company called QCEPT. And in my journey of learning new product development, working with new molecule, QCEPT has a very, very crucial role to play. This company was actually set up by a few researchers who were working in collaboration with University College London, trying to commercialize their ideas. So these research students had some ideas, had some patents, had some understanding of specific technologies, and they wanted to set this strategy moving forward to launch some products in the market using their platforms. So I learned a lot from them, and this is where we were doing a lot of preclinical work, a lot of solubility enhancement work, so that we can actually dose animals. And idea of dosing animal could be like using a suspension, solutions, nanoparticles, things like that, because you want to get maximum exposure in an animal model. The reason I'm telling you that, because I'm going to cover a bigger picture of product development today, right from preclinical to commercialization. And the idea behind is to give you sort of overview of overall drug development process. And I'm going to use some specific examples of antivirals today, just because we are in a situation where everybody is talking about viruses, especially the COVID virus. The rest of my journey in pharmaceutical industry actually progressed working on preclinical, clinical supplies, product launches, commercialization in different roles, different progressive roles. Most of them come from GSK, where I have led several product development campaigns, commercialization campaigns, and I'm currently working at Edifarm as a product development leader, looking after some of the product portfolio in injectable products. So that's just a little bit about myself, just to give you a flavor of why you should be listening to me today. Let's go ahead with today's discussion. Drug development process is a systematic approach, and it's called systematic because you need to know what you want to achieve from it. Okay, it is also important that you keep patients' safety in mind all the time, because any molecule you develop has to be safe. FDA will not allow another molecule in the market just because you want to launch it. So you need to demonstrate the molecule you're developing is actually safe. That's the first criteria that they will be looking at. Second thing, the new molecule has to be significantly different from the existing molecules or therapies, because FDA don't want just another molecule, another salt form of the same molecule who actually is going to do more or less similar things. So they don't want something which is already in the market and a little bit of changes in it. They want novel therapies, novel ways of working, newer mechanism to act, newer targets. So they usually promote scientists or pharmaceutical companies across the world to come up with new sort of solutions for the diseases or the issues we see in the world. So the process of drug development starts with sort of thinking about what you want to achieve. And this is the whole idea, because if you don't know what you want to actually target, it becomes very difficult. So the rational thinking, the target-oriented approaches must to have in a drug development process. So when I go to my objective for this presentation today, as I said, the idea is to give you a very high-level overview of what a drug development process looks like. Okay, we're also going to cover a few things around treatment options for COVID. We're also going to use some data from clinical readout to go through some case studies to see how we can apply scientific as well as a rational approach in the drug development. And also we need to acknowledge the fact that we don't know everything. As a scientist, the expectation is quite high, but the reality is that you will not know answer to everything until you've done extensive work, and that could take years and years. And that brings us to our timeline, because everybody wants this vaccine for COVID tomorrow, right? They want it today, actually. And what actually they don't realize, most of them, they don't realize that a drug development process is quite complex. A drug has to be tested in animal for safety. A drug has to be tested in first few, or let's say 8 to 10 healthy human volunteers to check its safety profile in humans. And then in the phase 2, it has to go through like more than 200-300 patients to prove its efficacy, whether it's effective or not. So there are different phases of drug development. In the phase 1 and preclinical studies, what we actually are looking at is understanding the pharmacokinetics and pharmacodynamics of the molecule, which is simply looking at what the body is going to do with the drug and what is the drug going to do inside the body. So thinking about the absorption of the drug, metabolism of the drug, distribution of the drug, and excretion of the drug. And what tends to happen is actually when you are dosed with a new molecule in your body, because it's the first time, your body actually don't know how to react to it. So it may create some sort of response to it, which may or may not be desired by the body. So this is where you may see some of the side effects. But early in the development, we are mainly focused on understanding the toxicity that could be associated with a new molecule. So toxicity could be as simple as this, like what is the effect of this new molecule on the liver cells? Okay, what is its effect on kidney? What is its effect in, let's say, if it's carcinogenic, if it has genotoxic sort of risk? These are some of the scientific terms we use when we're trying to understand the toxicity of the molecule. It simply means, is this new molecule going to be affecting the body, any organs inside the body? And if this effect is going to be sort of immediate effect, or this effect could take many years to sort of manifest. Okay, so some of the effects you may see could be immediate and others could be manifesting after a very long time. It's important to note these things because the expectation from everybody is that you want a safe medicine, but as you all aware that not every medicine is safe. Okay, when it comes to assessing the risk, the FDA usually looks for sort of benefit versus risk ratio, and most of the products that they give approval in the market, they try to justify why it is beneficial. You may have some sort of toxicity, you may have some sort of side effects, but overall, the molecule could still be considered safer. For an example, somebody who is suffering from cancer and he's going through a lot of pain and a lot of anti-cancer compounds given to the patients who are suffering from the pain can actually be killing the other healthy human cells too. I mean, cancer is abnormal growth of cells inside the body, right? It could be in any organ. And because this new molecule is trying to stop the progression of this cell or abnormal cell growth, it is also stopping the progression or growth or origination of new cells, of new healthy cells, okay? And as a result, your body actually do go through sort of side effects. But idea behind the treatment options is like, what is, in a cancer patient, how much life is left? Is he gonna survive for another 1 year, 2 year, 5 years? Is he gonna recover? So the risk is that he may not survive it. So what's the worst that may happen? He may have more pain, he may have more sort of side effects, but we take that risk, okay, with the cancer patient to say like, okay, let's try something more dangerous and see if we can still save him. He may see the side effect, but overall he may recover. Okay, that's the bigger picture to it. So a drug development process requires a lot of structured thinking, a lot of structured planning, because in a clinical study, when we are moving from, let's say, animal to humans, you don't know what dose you need to give to a human, okay? Because whatever is considered safe in animal may not necessarily be safe in humans. So you start with the minimum dose, and then you try to increase that dose of the new molecule to see if this is going to have sort of desired therapeutic response. For an example, if you start giving, let's say, 50 milligram of an active or a new molecule to somebody for fever, and if you don't see any improvement in terms of the temperature is not going down with 50 milligram, you will increase the dose to 100 milligram. And then you will go further up, like 250, 300, 500, whatever, right? And at some point, you may come to a conclusion that this is going to be very large dose, which may not be feasible to be dosed. Like you cannot give somebody like a big tablet, very big tablet, okay? So that dose titration, that fixing the dose also is important. And a lot of people don't understand. They feel like there's a drug, you test it in animal, and then it can be dosed in human. It's not. You have to actually fix the desired dose as well. And the study, clinical study, requires sort of a statistical approach to it, because if you're giving the medicine to somebody who is actually mentally thinking that he may recover on his own, or some people recover on their own. For example, if in COVID situation, the recovery rate is 80 to 90% naturally, people are recovering without taking any medication. So if you're developing a medicine for COVID, and if population is taking it, how are you gonna fix or how are you gonna actually understand that they recovered because of the medicine, or they recovered because of their own immunity? So these sort of challenges we come across all the time, because if people are recovering naturally, it becomes very challenging to understand how the new molecule is actually making their life better. Okay, so this sort of thinking approach and sort of structured design helps us and assessing the sort of risks with the new molecule, and also to see if they're going to be efficacious or effective in humans. So this slide here, just to give you sort of some information about what are the things that is done in different stages of the development of the molecule. The next slide is all about what if you get it wrong. Okay, there are a few occasions, and I'm sure most of you probably heard of thalidomide tragedy. In this tragedy, actually, a particular, let's say, sort of group of people who received this medicine, they actually had babies born missing limbs, missing their hands and legs. So this drug was taken by, let's say, some people who actually don't see any side effect in their generation, but the next generation who actually come about, they had the side effects. So thinking about the extent of the side effect, you may not see the side effect in this generation, but that happened in the next generations. About 10,000 children were born without limbs just because their parents taken this medication. Okay, another one, which is a French biotrial tragedy. I mean, about 128 healthy human volunteers were dosed with this new molecule, and suddenly what they had was few of the patients or few of the volunteers actually died because of multiple organ failures. And there are many other examples in UK, in USA. And what this sort of data suggests is that if we don't do enough preclinical studies, if we don't understand the data, or if we don't interpret the data in a correct way, it is likely that we may end up with a molecule that may do more harm to the people than benefit. Okay, and this is the whole idea. And this is where the drug development takes longer, because you want to be able to generate sufficient data before it's commercialized, used by millions of people. And sometimes data generated just on few hundred or thousand people may not be the actual representation of the whole world's population. For an example, your enzymatic composition in the stomach may be different to somebody else's enzymatic composition. Okay, maybe Europeans' body or body composition, let's say, is different to Japanese. Okay, and it is possible, and it does happen. So in those cases, without conducting clinical trial across the world in different parts of the world, you cannot generate sufficient data to demonstrate that the new medication would be safe. So think about it this way: when you're developing the medicine, you have to conduct sufficient studies in animals, at least 2 different strains of animals, and one has to be closer to human body, ideally a primate, chimpanzees, monkeys, things like that. And also you have to conduct the clinical trials in different parts of the world so that you can get a representative set of data. Okay, and what actually went wrong with these clinical trials here? One of the data which is taken from this French biotrial tragedy is that the molecule which was developed to sort of deal with the fatty tissues, fatty acids, the molecule was also binding to other receptors in the body, and one of the receptor was situated in the brain. And what happened was, when this molecule got inside the body, it basically blocked the sort of protein or enzyme, and because it blocked it, it led to a neurodegeneration and eventually death of the people who actually were part of the clinical trial. So if you don't understand your molecule completely, if you don't understand how this is going to be absorbed, distributed, metabolized, you may see this sort of effect. And this is the only reason why this extensive work is done, to understand some of those things. And how we apply this approach is what we're going to discuss today, that with all these learnings, we become more clever now, we're more evolved now, we have better understanding of how we should be designing our studies. But the risk is still there. The risk is still out there, because we, as I said, we would not know everything until we've tried this in a larger population. And that's just the sort of bread, not the bread and butter, but this is just the normal challenge a scientist have, actually. So when we look at some of the development approaches we have, just a second, let me just move the screen here. Some of the development approaches are quite empirical. When I say empirical, I mean they're still using age-old technologies where there is a molecule and that molecule is found to be effective in one of the indication. And what you do next is actually you try to change its structure by using a structure-activity relationship, sort of computational aided chemistry, different things, high throughput technologies. And you actually come up with new molecule, designing it. And that structural modification leads to a new molecule which may or may not be effective. And that is just the empirical approach to it. But what we're doing now is actually we going towards a more rational approach. Okay, there's a big difference on empirical approach and the rational approach. Empirical approach was let's design something and try. Let's look at sort of what may work. But rational approach is all about understanding the target first, understanding how we can design something that may fit our requirement. Okay, so this is more a structured approach to it. And when we actually are in this pandemic, everybody is looking into developing something more structured, but there are still few people, there are still few companies, there are still few, let's say, approaches out there which doesn't make much sense, but they're still out there. So we're gonna touch base on them as well today. So let's proceed. What I have next is, I'm sorry, just a second. As I said, the next approach is all about understanding your target, developing that understanding to understand your next target, which is this COVID virus, for an example. Okay, so in the structure of the COVID virus, what we've seen is actually this trimeric spike protein. What we also keep in an eye is the viral RNA, nucleic acid protein. I mean, these are few things that you please keep in mind. And when this virus came about, when Chinese government shared the genome with the rest of the world, all the scientists got thinking, okay, they went back to their drawing board. They started looking at some of the key structural weakness or strongness of this virus. And they started looking into their own molecules, which may actually work on those targets. So there were some molecules which were already in use, and there were other molecules that they could develop, okay? But we didn't have sufficient time to actually come up with new molecule. And that actually led to a situation where we started using what we had. I know a lot of you already have done a lot of study on this virus. And also, you know that this is not one of the most deadly virus we have. There are like many others, but it can spread very quickly. So I'm not going to go into that level of detail with this virus. But in terms of its structure, I want you to remember two things: the viral RNA and the trimeric spike protein. And we're going to touch base on them again later during this presentation. So please keep those two in mind. What we actually done after we identified the virus, we looked at its entry pattern. There are lots of theories of how this virus may enter your body. There are few data that's also suggesting that it is airborne. It means it can stay floated in the air, and when you inhale it, it can get inside your body. When it gets enter, when it's already in your body, let's say via oral route, via airways, and once it's inside your body, it's actually moving around and is going to find a particular receptor where it can bind. A receptor is an area where the virus can use its spike protein to attach itself. And what we noted in people who suffered from this virus was they had high concentration or dense population of this virus in their lung cells, pneumocytes. Okay, so that means the virus was entering in the body, but it was migrating towards lungs. And what we also seen that they were attaching to a particular cell site, which is called ACE receptor. Okay, so now we knew a little bit about the virus and also where it actually attaches itself and how it attaches. And rest of it is more about how it replicates. So a virus actually sticks to the body, enters to the cell, and what it does next is it goes through several rounds of transcription and translation to convert to another virus body. And this is a simple process of basically producing more and more viruses. And this process is very quick. I mean, virus can replicate like in millions, like within weeks. So this is where we see sort of, when you get first symptom, the virus concentration in your body may be different when you actually are in last stage. Okay. And in this whole entry process, the two things, one is the trimeric protein, and the cell receptor, which is ACE receptor, plays a crucial role. Once it's inside the body, there's very little you could do at that stage. So virus is inside, is replicating, is growing in number, and that actually challenged a lot of pharmaceutical scientists, like what we can do next. So what they actually did, and this is where I don't call this as a rational approach, but they had to do it because there was no alternative. So they started using some of the existing therapies which was already tested for toxicity in other indications. Okay, so hydroxychloroquine, for an example, is a medication approved for use in malaria, but is considered to be safer, and compared to other molecules, let's say, which are not tested in animals or humans, these were the best options to try. So different companies like Gilead proposed using remdesivir, which is now approved, hydroxychloroquine, Actemra, and there's like different antiviral combinations used for HIV. So this is all about giving a new purpose to the existing molecule, and this approach is called repurposing. And this repurposed medicine simply means this medicine are developed for something else, but can also be used in treatment of COVID. And the only reason pharmaceutical community started sort of using this or proposing use of it, because at this stage, we didn't have sufficient sort of knowledge on how to attack this virus. And we actually wanted to try some of the molecules which is already in the market and considered to be safe. And what we seen next was other companies, more innovative companies, they went back to their drawing board and they started their high throughput artificial intelligence approach to develop newer therapies. And we're gonna come to the newer therapies in a minute. But this slide here is to show you that some of the combinations, some of the new molecules, or some of the existing molecules currently being used, they're only being used because they were considered safer, and in comparison to molecule which is not tested in humans, I mean, these were the best options we had. And what we actually started sort of generating data on was looking at how these molecules going to work in this virus. Okay, so for an example, if we look at remdesivir, it is basically a nucleoside analog, and what it does is actually it basically places itself in the RNA structure of the virus, and by doing that, it stops its replication. The combination of antiretrovirus like lopinavir, ritonavir, they were actually stopping the protease enzymes or reverse transcriptase enzyme, things like that. So they were stopping different enzymes so that virus cannot reproduce. Hydroxychloroquine was kind of affecting the endosomal pH environment. And all medicines were working by different mechanism and eventually killing the virus or sort of reducing their replication rate. One thing that is mentioned there, which is monoclonal antibody and convalescent plasma, I'm going to come to that because that is where our approach is more specific. So far, what we've tried, like different drugs which were existing and those repurposed medicines, it's like use what you have, okay, but also keep thinking about things which we can develop. And monoclonal antibodies, convalescent plasma, they are actually the future of therapies, and they are very selective in their approach. So we're going to come to that. And that approach starts from our immune response, okay? Those monoclonal antibodies, they are actually produced by our own body. So when a new virus enters our body, the body treats it as a foreign material, and very soon our blood cells, they start going around it, trying to identify it, trying to touch it, to see some sort of structural weakness so they can attach themselves to it. The whole idea is to remove the foreign material as quickly as possible. That's body's approach to it. Okay. And in the process, they release different things. They can actually approach the mast cells, and mast cells can release different sort of histamines, and they can lead to inflammatory response. So you may feel feverish, and different cells does different thing. But the whole concept of innate immunity at that stage is to somehow neutralize this new foreign material in the body. And then there are T cells. These T cells are actually sort of two-way sword, because they can actually work in your favor and sometimes against you. And what we see in COVID patients, that a lot of them died with pus built inside their lungs. And how that pus actually developed there and how those lung cells died. There is a theory that when this virus entered in the lungs, the whole body got freaked out. It went to sort of a stage where it started releasing every possible thing it had to cope up with the virus, because there were just so many of them. So they started releasing a lot of histamines, cytokines, these sort of hormones and these sort of materials, let's say. And they actually led to attraction of T cells or natural killer T cells towards them. And these killer cells were destroying our own cells. Okay, think about it this way. When China first reported patients in their province, they reported people dying with sort of blood in their intestine. The reports in Italy, where they saw a lot of deaths, they were saying there were like multiple organ failures. In US, they generated data to say that there's a significant amount of kidney failures in those patients. So different part of world, when you look at the clinical data, they were indicating different things. But one thing that was common amongst all was that the body was responding in a very, very sort of strange way to this virus. Okay, why the organs were failing? So one of the theory has it that these mast cells or cytokines, when they actually released in your blood, because they want to protect your body, they can also cause vasoconstriction, and that vasoconstriction can actually lead to sort of slowing down of blood supply to different organs, because your blood cells or the arteries and veins, they actually who supplying the blood, they actually got constricted, and now they cannot actually supply the blood to the organs, and eventually those all organs died. Other theory is that your lung, where the oxygen exchange takes place, obviously your body need oxygen to survive, because the lung was full of pus, and obviously the natural killer T cells were destroying the cells. There was not much of exchange of oxygen in the cells in the alveoli. As a result, the organs failed because they lacked oxygen. Okay, so there are multiple theories suggesting that the organ failure was happening because of multiple reasons, but the bottom of that failure was these sort of responses. One response that is slower, this is called adaptive immunity. And this is adaptive immunity because your body is still studying this virus, okay? And in the process, it's trying to develop some antibodies. At the beginning, the concentration would be very low because they're still sending some messenger out there. That messenger is trying to see where this virus is, trying to find the weakest spot in it. And that way, this adaptive immunity takes slower. The antibody, specific antibody, which can attach to the virus and can neutralize it, can take some time. And this is why people or the government bodies and hospitals have been recommending people to stay in home and observe the situation for at least 14 days, because it can take up to 14 days for your body to develop those antibodies. Okay, and that is what your immune response is all about. And now scientists are looking of ways how they can actually invoke that immune response in people without using the virus. Because if your body already has this, let's say, antibody, if this virus comes in your body, then your body can quickly neutralize this virus. And that is the whole idea of developing the vaccines. The vaccines are actually nothing but sort of materials. Once injected in your body, it creates sort of immune response, and that immune response can actually lead to generation of loss of monoclonal antibodies or antibodies. Okay, these vaccines could come from different sources. Sometimes, I mean, most of the old vaccines, they actually are made up of dead or attenuated, which is weakened version of the virus. And when they dose in the body actually tries to react to it and generates the antibodies. So the newer approach, some of the newer approaches is like find the genetic code. And this is where this S spike protein is important, because that is what we believe causes this immunogenic response. Okay, so anything that is closer in structure to this spike protein, if it's injected in your body, it could be using a recombinant protein, it could be using a nucleic acid structure, which can eventually sort of convert to this spike protein and can lead to this immune response, will give you sort of protection. And what we were trying with repurposed medication is we are trying to kill the virus which is already inside the body. Okay, but with the vaccine, what we're trying to do is a preventative measure, because once you have the vaccine, you have the antibodies to fight this. So there are two different things. One is about killing the viruses which is already inside body, which is called treatment, and the vaccines are preventative measures. So that's the basic difference. And a variety of obviously vaccines are being trialed. They are going through different stages of trial at the moment. We've seen some promising data with the vaccines that has been studied in Oxford last week. So there are lots of things that's happening, and the vaccines may be the way forward for ultimate control of this virus, but it may be a while before we see a successful vaccine coming. Okay, let's look at some of the clinical data so we understand what is happening when we're taking those sort of medications. A lot of people started talking about treatment options. A lot of pharmaceutical companies started using the combinations they already had. And what I have in this clinical readout is a combination of antiviral, which is lopinavir and ritonavir. And when this combination was given to patient population, and when the same patient population received a placebo, which is nothing, just sort of sugar pellets. Okay, so you're trying to study if your medicine combination is actually better than no medicine at all. Okay, so one set of population receives this medicine, other set of population receives no medicine, and when they actually looked at sort of benefit of this treatment, it wasn't much different to not having this medication. And that is a bit discouraging, because we thought that antivirals may work. Okay, but this data is suggesting us that it's not better. Actually, it's not better than not taking medicine at all. And the same data goes with the hydroxychloroquine as well, because the hydroxychloroquine was considered sort of treatment for it. But the data suggests that your sort of recovery, sort of probability of living virus-free is not significantly different if you're not taking any medicine at all. So this sort of data is important for us, and being obviously sort of brain of the society, responsibility lies on you people and scientists to sort of look at this data carefully, interpret this data, and then come up with sort of ideas. So this data may look discouraging, okay, but what is also indicating us that maybe this particular combination of medicine didn't work, but when we combine this medicine with others, it may work. Okay, so hydroxychloroquine is combined with azithromycin. The lopinavir is also being tried with ribavirin. So like now the scientists are actually looking at different combinations, okay, different mechanism of attacking the virus. And by doing that, because obviously these molecules are already in the market, they are safe, whatever the development path we're taking now is actually not causing much of delay because we already are using this. Let's use this in clinical studies. So there are like over 70 clinical trials running in the world at the moment, looking at different combinations as well as new vaccines. Okay. What we also have to keep an eye, obviously as a scientist, is that when a company is trialing, let's say one molecule, because remdesivir is approved in the market now, sometime they may have other product in their pipeline, which could be equally effective. And this is a case study of the clinical data where Gilead's own product, remdesivir, was when compared with their own molecule GS441524. Actually, what they spotted was the remdesivir takes 7 different metabolic steps to convert to the active form, whereas GS441524 can convert in just 3 steps. So basically, quicker the conversion, quicker the active form is available to kill the virus. Okay, but remdesivir was taking longer. When they actually compared these both molecules in an animal study, what they noted was there isn't much difference in how actually they are sort of improving the life of animal or killing this virus. That means both of the therapies are effective, but one can convert quicker than other. Okay, and it has, the new molecule also has a better solubility and lower toxicity, which means you can dose more quantity of it, and more quantity of this drug when available inside the lungs can kill the virus quicker. So simply means the other molecules in the pipeline of the existing companies, existing therapies may also be effective. Just because we don't have enough clinical data, we are using the one which is sort of suboptimum, but is still doable. Okay, so having said that, you know, the remdesivir is already in use. There will be some sort of studies to look at sort of increasing its dose to make it more effective. And that's the whole idea behind this data, actually. So let's look at some of the normal challenges we see when we are in the product development stages. I mean, just looking at previous examples, bioavailability, which is amount of drug available inside the body and the rate of its absorption. What is actually indicating us that a lot of drugs, they cannot make to the market. I mean, the ratio is quite bad. 10,000 molecules goes to animals and only 1 molecule can make to the market. So the ratio is quite bad. But what happens to the rest of the molecules? I mean, a lot of them fail in the stability. And this stability is important because when you develop a product, you know, you have to formulate it, store it, transport to the many parts of the world where the temperature may be different, the humidity may be different, people may be storing it outside the fridge, inside the fridge, different places. Okay, so it's important to study stability and also bioavailability if it's going to be working at all. Sometimes there's a risk of safety. As I said, the safety is important aspect to it. So every single molecule you're going to develop will have to show that it's safe and efficacious. Cost is also important factor because not every pharmaceutical company can afford millions and billions of dollars to develop the product. And also they don't have luxury to spend 10, 14 years to develop the molecule. So these are some of the key challenges pharmaceutical companies are facing. And to overcome them, they rely on scientists to come up with sort of novel ways and smarter ways. And this is where your technical capability, your smartness comes handy. Okay, so these are some of the things which we see every now and then. These are the reasons why not every molecule can make to the market. And other things that we do during the product development is to study the product, its physical and chemical nature. So we talked about its biological performance in animals and in humans, but equally important, you need to understand the molecule itself physically, like what's its purity like, what is going to do when you expose it to moisture, how its particle size looks like, if it's going to be flowing well, not flowing well, if it's going to be converting to a different form over the period of time. So these are some of the key things which we actually study with the small molecules. Okay. What you also see here is solubility data in different pH environment, because in the body, your stomach has a different pH than your intestine. That simply means the acidity and alkalinity of different parts of the body is different, okay? And that can actually affect the absorption, the solubility of the compound. So this data actually is helpful for us to understand how this drug is going to get absorbed in the body, okay? Let's look at some of the large molecules. Large molecules are nothing but sort of antibodies, protein peptides. The problem with this sort of antibodies is that they lose their stability and activity very quickly when they're exposed to sort of high temperature, different ionic concentration. Even simple stress of shaking the vial can also affect their stability. And we use a large variety of bioanalytical tools to understand their structure, a variety of them, more than 20. So in order to develop all those 20 methods, generate sufficient data, interpret the data, and find conclusive sort of data from it can take like months and months and months and months, okay? So the drug development process is not just the bioperformance, it's also the physical chemical characterization to these molecules, and the variety of analytical tools that you may have to use may surprise you. So a lot of work that is done in the background that not many people are aware of, but these sort of studies are important to provide sufficient data about efficacy, safety, stability of the compound itself. Once it's actually entered into phase 2, we're thinking about commercialization of it. So for an example, for manufacture of COVID vaccine, let's say, the Bill and Melinda sort of group has already identified 7 different manufacturing companies across the world. And in India, there are 2 sites which are also identified, Serum and as well as Cipla. So what is happening here is that you've done your physical chemical characterization, you've done your analytical method development, you know your formulation now. Formulation is basically just the mixing of different excipients with the active to make it easy to dose. So what's happening here is that you're thinking about other things now because you are in manufacturing domain, you are looking into commercialization of the product. So how are you going to launch this product? What is your regulatory strategy? Because different companies have different regulators and different regulator may ask different information. So before you can launch the product, you need to define the target market, the regulatory requirements. When you are manufacturing, you need to understand what are the critical process parameters which can affect the performance of the product. You need to have sufficient analytical capacity to sort of analyze the product. You need to have sort of cleaning in place process to understand if you can clean this product at all, because you're not going to be manufacturing just one product, you'll be making multiple products. So if you don't clean it effectively, it may contaminate other products you manufacture in the same facility. So a lot of things that you need to understand before it can be commercialized. Think about it this way: when you're launching the product, all of your product is kind of coming from small scale batches. And when you are going to the larger scale, you basically sourcing it from other companies because you probably don't have the capacity to manufacture, let's say 300 kg of the material, okay? Or let's say probably 1000 kg of the material. So you're sourcing it from China, India, places like that. But them places will not necessarily have the same equipment, same capacity or capability. So your product, which you tested in human, may be slightly different to what you receive from a different supplier. So understanding the effect of those smaller changes between one supplier to another supplier can also cause sort of issues during the manufacturability of the product. This is all, the idea behind this slide is to give you overview of things that can go wrong, the things that takes time, the things that actually is done to basically commercialize this product. So we done the testing in animals, we done the testing in humans. Now we're trying to sell this product to the market because we thought, or we have sufficient data to say that this is safer now, okay? So on the same process, I want to give you a practical example here that when you going from a smaller scale, that variability not necessarily come from one supplier to another supplier. It can also come from the same unit when you are going from a smaller batch to a bigger batch. What we have here is the PMA25 on the left, which is a small granulator, let's say 10, 20 kg. And then you have PMA65 in the middle, and then you have PMA150 at the end. And what these three different granulators are sort of indicating us three different batch sizes, okay? Because when you're manufacturing it, you're manufacturing this in batches of 50 kg, 100 kg, whatever. Like when we actually are scaling up, when we are going from 10 kg to 200 kg, 500 kg, the performance of your product can also change. For an example here, when we were trying to go from a smaller scale to larger scale, we were seeing slower rate of dissolution. And that dissolution was related to change in granules property in the tablet. And that change in property was related to how much water that went in, how much weight massing was done to actually generate that granule. So granulation process is part of tablet manufacturing here. And that granulation process is dependent on some variables, and some of the process variables are actually the amount of water you're adding, how you're drying it, the weight massing time, how much time you allow these granules to come in contact with water, things like that, okay? What we were seeing here is that when we're moving from a smaller scale to larger scale, the performance of the product was changing. So as I said earlier, that your performance can actually change, okay? And because your performance can change, it may ultimately affect the bioperformance of the product inside the human body. And how do you make that assessment? So we cannot take every single batch to clinical studies. Once you have commercialized product, or you are in process of commercializing the product, you need to have some sort of analytical tool or analytical method to check the consistency or reproducibility of the process. Let me repeat it. Consistency and reproducibility, that every time you manufacture this product, it will have the desired therapeutic outcome every single time, every single batch. And the only way we can do it is by developing some sort of in vitro methods. In vitro methods is like the testing that is done outside a human body. As I said, you cannot dose every single batch of product you manufacture to humans to understand if it's going to be performing equally well or not. So what we do here is we use in vitro techniques of small basket of water, let's say. We drop our tablet there and we look at its dissolution rate. Why are we looking at dissolution rate? Because when you take a tablet, it takes some time to dissolve inside your body, right? And that dissolution here or solubility here can actually delay or quicken the rate of absorption. And the type of profile you get inside your body could be dependent on the concentration at the time it's released. So basically you're looking at, let's say, 80% of the drug release in less than 30 minutes or 100% drug release in an hour, okay? That is sort of, let's say, your specification for this particular product. And that specification is based on your performance inside the body, because when you've done this study first time in human, you already know that within like 30 minutes, you start seeing some effect, okay? That effect is happening because your drug is available in your blood now, and that blood is actually distributing this molecule across the body. And that is how you're seeing this performance in vivo inside the body. And in order to replicate or mimic the similar performance for other subsequent batches, you're testing every batch in a dissolution basket, looking at the rate of release of the drug every 30 minutes, every 45 minutes, or let's say every 60 minutes, okay? What we saw during our development process for products, that your manufacturability may be different, for a number of reasons. Sometime an operator may make a mistake. Sometime an operator during the manufacturing can add more water, less water, more weight massing time, less. There may be variability to manufacturing process. Sometime your manufacturing unit, let's say your granulator is gone old, and its performance has changed from what it started 10 years ago. So there will always be some sort of variability in your batches, which is what we've seen here, that for, the two batches with slightly different water content, slightly different weight massing time, But when they actually tested inside the dissolution basket to predict the bio performance of it, it wasn't significantly different. And that is good because that is indicating us that your product is so robust, is so stable that a small amount of changes in your manufacturing does not affect its performance significantly. So the whole idea is to actually have a robust product during the commercialization, which can consistently deliver the right dose so that we can get the right desired therapeutic response every time we manufacture this product. So what I've done today is actually to give you sort of a little bit understanding of preclinical process, clinical process, manufacturability complications, some of the ideas we're using to develop sort of therapies for COVID. And that actually takes me to my last slide, where I believe that, now we have more appreciation of drug development process. What we also know that this is a continuously changing environment, continuously evolving, because every day we get new data. Every day we have slightly different understanding, looking at scientific publications, clinical data, and then actually we try to adopt our approach, change our approach, and that way we will find the cure. And for now, because our antibodies are the one fighting against this virus, we are the cure. Okay, that's the message here. And I'll be very happy to take any questions if you have.
Sir, actually, I have one question, sir.
Sorry, who's there?
Prashant here, sir.
Hi, Prashant, go ahead.
Actually, sir, just I wanted to know what is the principle behind considering hydrochloroquine? Is WHO has had been assigned any repurposing guideline with remdesivir and chloroquine? Because many of the clinical trials study I have gone through, but I didn't find any subsequent and a specific idea by which factor and by which ideology MCQ and remdesivir has been repurposed and it is going to be prescribed across the world.
So actually, I don't know if you noticed or not, in one of my slides, what we've seen actually is remdesivir was already actually in use for treatment of, let's say, certain viral diseases, okay? What we've seen is that the target site here on this particular virus has the same enzymes which remdesivir can attack. And that is the same concept for hydroxychloroquine as well. We already had the data that every virus is different to each other, but there are some similarities to it. So the type of mechanism they can both work at could equally be applicable for COVID virus. And that was the idea behind it. But interestingly, when artificial intelligence approach was also used, and again, your artificial intelligence is not the solution, it's the tool, right? And that tool actually, when you fit in your information and that actually does its own algorithm to come up with sort of solution, it does indicate that some of these molecules could be effective. So the repurposed use of some of the existing molecules wasn't accidental. They already knew some of the route or mechanism where this can affect or actually work on. And they also demonstrated using the AI technique that the approach they've taken is actually a right approach.
Fine, sir. One more question I have, sir. MCQ along with azithromycin is being prescribed, but azithromycin is also not a right therapy to counter the COVID-19 because it is having a resistance capacity ahead. So how could we overcome this resistance? Because many of the antibiotics is being prescribed with some other molecules. So can we expect a less a higher efficacy rate with other antibiotics along with hydrochloroquine?
Actually, the combination of azithromycin is obviously trial because the pneumonic condition we've seen in some of the patients. So this is an antibacterial, okay? The antibacterial doesn't work against virus. So the concept is not to kill the virus, but to help hydroxychloroquine to minimize the damage inside the lung. So once your lung is weakened, it's basically showing symptoms of pneumonia. And that pneumonia can actually be sort of had or aided with azithromycin combinations. And that is why some of the antibacterial combinations are being used. Yes, azithromycin may have resistance to certain bacteria may have resistance to azithromycin, but not all of them. And as I said, the repurposed medications are being used only because they actually are proven sort of safe, and other molecules which may come in the market, which may be more effective, are not there just yet.
Thank you so much, sir. Thank you. It was a wonderful information. Actually, I had not been interacted so early, so it is really a very informative session. Now, if anybody want to ask, hand over to other delegates also.
Thank you.
Good afternoon, sir.
Hello.
Hello.
Hi.
Hello. Good afternoon, sir.
Good afternoon.
Thank you so much, sir. It's a very for me, it's one of the best session I have attended during this lockdown period.
Thank you so much. This is good.
Sir, one question for like Indians, lots of people are talking about if anybody is immunized with BCG vaccine, so they have a very less chance of getting infection with COVID-19. What could be the reason?
I mean, that is the same concept. We said that if somebody has received influenza vaccine, they probably have less chances to actually die from COVID. The same concept went with the HIV patient. Like HIV patients are already taking antivirus combinations, so they will probably not get the viruses, right? But in order to make anything conclusive, or let's say sort of any sort of proposition or any theories have to be backed up with clinical data. At the moment, we don't have sort of sufficient data to say if this BCG vaccines, and because you need to know who is taking this medicine, what stage they're taking it, and then how this actually BCG vaccine is neutralizing or BCG has generated sort of antibodies which are neutralizing this virus. So just saying that it is effective is not sufficient enough. You need to be able to demonstrate that practically, like, okay, BCG actually produced this particular antibody, and this antibody is actually neutralizing the virus. We don't have the data. And more we study this sort of process, more the data we will have. So it's not just the BCG, is the flu vaccines, is the antiretroviral combinations. There are several things. Some people are also using anti-anthelmics. So there are lots of medicines under clinical trials for different reasons. And it will be obviously matter of time when we will have more clarity on which is probably more effective and why they are effective.
Okay, sir. Thank you so much.
No worries.
Prashant sir, any more questions?
I think there is no more question. You may proceed ahead.
Okay.
Thank you.
Thank you so much, sir, for such an informative session. Arkajan University will be very grateful if someday you visit our university. And now I would like to request our Dean, sir, Dr. Jyotirmaya Sahu, to hand over the lifetime certificate and certificate of appreciation from the university to Mr. Bhupendra Kaushik, sir.
Thank you.
Thank you, Bhupendra Kaushik ji. A wonderful session with you, with a lot of information. We are proud to be with yours for a wonderful session, and we expect your benign presence in future whenever you will be to India.
Thank you so much, Jyoti sir. Thank you so much. This has been great, actually. You guys been very disciplined, paying attention to, listening to it, and I hope you benefit from it. And I'm really grateful to everybody who's been part of this, making this organizing event, and particularly to Prashant, who's been working really hard. Thank you so much, all.
So as a token of appreciation, a virtual certificate is prepared by AIPS and Arkajan University IQAC School of Pharmacy.
That's
so
amazing.
Please, sir, accept it.
Thank you. Thank you so much. I will. Thank you.
I also request Abhishek to please showcase a lifetime certificate of Honorable Bhupendra Kaushik sir. can we have a huge round of applause to Kaushik sir? Thank you.
Thank you so much. Thank you. This is amazing. Thank you so much, Prashant.
Thank you, sir. Thank you. Thank you for thank you for your time and information. And we would love to have you for a wonderful session in future ahead. So from the University Corner and on my behalf, once again, I would like to thank you. Thank you so much, sir.
Thank you so much. Bye now. Have a good day. Take care all.
Thank you. Thank you. Thank you. Okay. Thank you.
And this is the right time, Prashant. I think our participants are also there. Valuable participants are there from different corner of India and the world.
Yeah.
We should have a huge round of applause for them.
Yeah. Sir. Yes.
Please,
Sir.
Yes. Please, Prashant sir.
Sir, we may close the meeting. We may close the meeting.
Yeah. So everybody, we will meet tomorrow at 10:30.
Thank
Right,
you.
sir?
Thank you, one and all. Yes, sir. Thank you. Thank you, one and all.